Hi everyone, today we're going to talk about cell theory and microscopy, so let's get started. First, we're going to start with the introduction to cell theory, which states that all living things are made up of cells. Cells are the basic unit of all life, and also all cells come from pre-existing cells. When we look into the history of cell theory, it starts with Robert Hooke in 1665 when he first named cells. Anton van Leeuwenhoek improved microscopes, and because of his this improvement, he started seeing microbes through microscopes as well. In the 1800s, Schleiden and Schwann formalized the cell theory, and finally, later on, Rudolf Virchow added all cells come from other cells, meaning cells need other cells to be pre-existing to form new cells. So we started adding some modern additions to the cell theory, such as the idea of energy flowing within all cells. Cells also contain hereditary information contained in what we call DNA. All cells also have the same basic chemical compositions. We use a tool called a microscope to look at cells because they're so tiny. Microscopes are used to magnify and resolve small objects. We look at two things when we're looking into a microscope, and that is magnification and resolution. Magnification means to make an image larger. So when an image has or is being looked at through low magnification, we're looking at it from a farther viewpoint so that all the cells look a little smaller. When we're looking at high magnification, we're basically zooming in to make the image larger. When we're talking about resolution, we're talking about clarity of detail. So when we're looking at low resolution or objects through lower resolution lenses, we're not seeing the image that clear. It's kind of blurry. But when we're using higher resolution lenses, we're creating an image that is clearer or higher resolution. There are several types of microscopes that we can use to look at small objects such as cells, and the main ones are light microscopes and electron microscopes. Light microscopes use light to look at cells, and they're very useful for looking at living cells. When we're using electron microscopes, we're shooting a beam of electrons at whatever we're looking at, and normally electron microscopes create an image that is higher resolution. The con is that electron microscopes can only be used on dead samples. There are two types of electron microscopes. The first is a scanning electron microscope, and these microscopes are very useful for creating 3D surface images like this one. But we also have another electron microscope called a transition electron microscope, and they're useful for you looking at internal details of a cell like this image. In summary, cell theory states that all life is made up of cells. Cells are the most basic unit of life, and all cells come from pre existing cells. Some modern additions to this theory include the energy flow between cells, all cells containing DNA, which is hereditary information, and all cells containing the same basic chemical composition. Microscopes are great tools to use to study cells and see them in detail. So in the next video, we're going to talk about prokaryotic versus eukaryotic cells, their similarities and differences, and how the differences in structure affects the differences in function. That's it. Thank you.